and not the 3 base that we done in the past. Because what we've done for you in the past is just the center of the ribosome, and it turns actually 3 areas to the ribosome called the A, P, and E sites. And we'll get into that in just a minute. But first, all right, so what we're doing here, let's get rid of all this. Oh, it's not focused at all. All right, what are we doing here? So it's bound ribosomes, size proteins primarily for secretion, okay? Meaning attached to the ER. So these are ribosomes on the rough ER. Bound ribosomes synthesize proteins primarily for secretion or for use in lysosomes. These are the organelle that are responsible for digestion. These are found only in animal cells. All right, these are considered the floating stomach of animal cells. Or for use in lysosomes, while free ribosomes, there are ribosomes that float around in the cytoplasm. They are not attached to the ER, or we call them free. Free ribosomes synthesize protein for use primarily within the cell. Okay, so we need to draw ourselves the classic drawing of the cell with the notch in the corner. So that's going to be, let's put the notch down here. Boom. Hey, that's not bad for me. Wrote on the table a little bit though. All right, so this would be 2E. Okay. Oh, you have the nucleus. And of course, inside of the nucleus is DNA, of course. Okay. And then we can zoom in on this. Mm-hmm. And then when it comes time to express a gene, the gene is read by RNA polymerase and through a process called transcription. Okay, and that forms a molecule of messenger RNA. I'm hoping this is just super straightforward. So this messenger RNA is going 1-2 places. Okay. It could be going over here out into the cytoplasm and be read by a free ribosome. We will come back to that messenger RNA later. So this is messenger RNA. So what we're going to do first, let's go like this, and then a little more here. Something like that. And then, so this is going to be your rough ER. Okay. So then sitting on the surface of this rough ER are the ribosomes. Now, it actually turns out that it's only the small subunit. That ribosome should not be there. It turns out that it's only the small subunit that's on the rough ER, but you will find out about that when you take cell bio. Such. It's really only the small subunit messenger RNA comes in, the large subunit cap comes in and locks it in. So the messenger RNA then goes here, like such. And it is read by the ribosome here, what we call an attached ribosome. Attached because it's attached to the rough ER. Okay. The ribosome then reads the messenger RNA in groups of 3 called a codon. The codon then bonds to the anticodon on a transfer RNA, and you bring in amino acids in a very specific order. And this is what's called a primary protein. Okay, that protein is based on the order of the amino acids. Then this protein starts to twist and fold into its secondary conformation. When you talk about protein shape, it's called conformation. And so that is the secondary conformation, where if it's a spiral, it's called an alpha helix. And if it goes up and down, it's called a beta sheet. And then that protein starts to twist and fold back on itself into a three-dimensional shape. We call it globular, because when you put these proteins in water, they look like a glob. Okay. And then this protein is packaged, this tertiary protein is packaged inside. Oh, I didn't leave myself very much room for a 3. Let's squeeze that in there right there. I want green. Let's do that. Squeeze it in there. There we go. Okay. And then this vesicle with its tertiary protein pinches off. I'm going through this relatively quickly because this is core 2. If this doesn't make sense, you need to go back to protein synthesis in core 2, topic 6 or 7 in core 2. And then we get into the Golgi body, such. Such. So then you have your tertiary protein here, like such. And then you will have other tertiary proteins. What do I want? Let's do purple. I don't want green, that's redundant. Maybe pink. Pink might not show for you. Let's go for a blue. Let's go like this. I should test that other color. On your screen, does that look purple and that looks blue? Is there enough of a difference for you between those two colors?
Yes.
Yes, excellent. So then we'll go over here. Boom. So then these are tertiary protein, tertiary protein, tertiary protein. So what happens inside of the Golgi body is there's editing of the proteins. And these tertiary proteins are then bonded together. There's a lot of control here. There's a lot of biochemistry that goes on in here, tremendous amounts. I am speaking in very broad generalizations here. And when I just, it's just like when you edit the messenger RNA with the spliceosomes here, we're editing the protein. People have full PhDs in that. So here is your quaternary protein. Like such. And so this will be a quaternary protein, and then this will be a quaternary protein like such. So then this is quaternary. This, whoops. Oh, I did it in green. Rats. Quaternary. Quaternary. That's all right. You guys can handle it. So then we'll go red, red, red. Then we'll go blue, blue, blue. It is. See, I give Em a hard time because she's going to color with children. Elementary school teachers, all you do is color. And then here I go with my big fancy science stuff and color.
And your crayons.
And my crayons. It's okay. I'm comfortable with my crayon work.
You've got crayons.
Yeah, that's all right. I'm okay with that. Okay, so this should be a total review diagram. If it's not, then get back to core 2. But what this diagram shows you is, oh, and I didn't do translation. Good grief. Here's translation here, right? So transcription in the nucleus, translation on the surface of the rough ER, primary, secondary, tertiary protein bonded to other tertiaries to make a quaternary protein, and ultimately attached ribosomes, all right, synthesize proteins primarily for release from the cell or excretion. I'm not saying that there are not exceptions to this trend, but the overwhelming majority of proteins that are produced by attached ribosomes leave the cell through secretion. Okay. So you then have the ribosome here that attaches to the messenger RNA in the cytoplasm. Since this ribosome is not attached to the rough ER, we refer to it as a free ribosome. So free ribosomes synthesize, and I want to take out that messenger RNA. I don't like where that is. You go away. Free ribosomes synthesize cytoplasmic proteins. So this is easy. Primary protein. Secondary protein. Tertiary protein. I haven't read that one yet. Let's go. So this would be primary, this would be secondary, and this would be tertiary. This would be primary, this would be secondary, this would be tertiary. So what does this imply about the conformation of almost all cytoplasmic proteins? Almost all cytoplasmic proteins have what kind of protein conformation?
Tertiary.
Tertiary, you bet. Of course, there are some exceptions, but the overwhelming majority of cytoplasmic proteins are tertiary. I'm going to put this up. So this is 2E. And so what you want to do here is draw this out and then discuss and describe what's going on. And then if this is review, fantastic. And if it's not, then go back to core 2. Just like that. It's so pretty. Except for the green 4, right? That's your yellow. Or yellow, what's the name? Red. Whatever. Go. You okay?
Yes.
Okay. Then, as this protein leaves, though, some of these proteins stay embedded in the plasma membrane, okay? And you know that the plasma membrane has areas that are polar and areas that are non-polar. And so I want to talk about how this protein here, how this primary protein has areas that are polar and areas that are non-polar. So make that go away, right here, and let's continue on question 2E. How'd you guys like all the wind yesterday and the hail last night? Yeah. I was hoping for some good what's the word I want? Lightning and some thunder. You guys ever been in a really good lightning and thunderstorm?
Yeah, I think so.
Yeah, where the flash of lightning hits and then within like half a second, boom, you get the thunder. It's really, really cool. Not dangerous at all, as long as you're not standing outside and get hit with the lightning. The lightning, you'll never know, right? It's not like you'd be like, oh, the lightning's hitting me. That's not the way it works. If it hits you, you're with us, and then you're not. So at least it's quick. But a true, really good thunderstorm is, I find it super exciting. It's really dynamic, and just the raw power of it, I think, is super cool. All right, so I'm going to pick 2 different colors. So I like this color. What is it? Blue green? Interesting. It's kind of an aqua. There you go. And I'm going to use this color for polar amino acids. And I'll pick a dynamically different color for nonpolar amino acids. Now, something that we don't talk about very much in IB is the polarity of the amino acids. Some are polar, some are nonpolar, some are acidic, some are basic, some are neutral. We don't talk about them almost at all. This is just about the only time that we hit the polarity of the amino acids. So when we draw a series of amino acids like such, okay, And then you'll see me label this as a primary protein, like such. Understand that, of course, these are all individual amino acids, okay? And so some of these amino acids are polar, and some of them are non-polar. And so as you look, I'm going to then change the model like such. And so you might have a series of polar amino acids here, and a series of polar amino acids here, and a series of polar amino acids here. Oh, there's my original. There it is. And then you might have some non-polar amino acids here, and non-polar amino acids here. Now, don't forget that this is a primary protein. Okay, or of course, polypeptide. Now, as this protein starts to develop into its final form and get into it's going to twist and fold on itself, yes?
Yes.
Okay, so what we need to draw, let's draw ourselves a phospholipid bilayer and then leave a big old gap like this. And I know I'm not drawing these phospholipids very close together. They're really not this far apart, of course. And then we've commonly drawn a channel protein like such, right? Whether you're talking about a sodium-potassium pump, whether you're talking about a sodium channel, a potassium channel, a chlorine channel, whether you're talking about the membrane protein that insulin bonds to and glucose then travels through. This is some kind of transport protein. When you take cell biology in college, there's a difference between a gate and a channel and a there are proteins that transport materials in different ways in different directions is how I'm going to leave that. But then understand that there's a polarity difference in the membrane right at the base of those heads. How is this part of the membrane different from this part of the membrane? What's the polarity of this part of the membrane with the glycerol heads?
Polar.
Yes, it's polar. Excellent. So this area here is polar, and this area here is polar. Okay. However, the center of the membrane here, what's the polarity of the center of the membrane here?
No polarity.
Non-polar. Nice. So then does it make sense that these different areas of the protein also have to match the polarity of the membrane that these proteins are embedded in? Does that make sense?
Yeah.
Okay. So then what do we pick? For polar, we picked the blue. So that means this area has to be polar. And this area has to be polar. This area. So this is where the folding and twisting, the conformation of proteins is incredibly important. Okay, because if that protein doesn't fold properly, then it won't properly sit or be seated in the membrane and it won't work. And then, now here's what's also cool. Let's say that this is a transport protein that handles glucose. Well, glucose is a sugar that's polar. So then what does the side of this channel have to be, since glucose is polar?
Polar.
Yep, it also has to be polar. So even the channel through the protein has to be either polar or non-polar, depending on what the transport protein is transporting. Whereas this area out here would be non-polar. So there's a lot more chemistry to this when we talk about the proteins folding and going out of the cell or being embedded in the membrane. there's more to the folding of the proteins than originally meets the eye. So then the glucose would then come up here, because of course glucose is a sugar and therefore it is by definition polar. You good?
Yes.
Yeah. Anyone still need this? That's a pretty good little picture. That's cool. All right. Here. Yes.
Can you please repeat the section you said? My internet cut off.
Could you ask me that one more time?
The part you just explained, can you repeat it? My internet was cracking.
Sure. About the channel on the inside here. I think that's the last thing I talked about. I think. All right. So center of the transport, the actual.
Yes.
Okay, got it.
Channel.
The actual channel where the molecule travels through. Okay, depending on what you're transporting through the protein, determines whether the lining of this channel is going to be polar or non-polar. The lining of the channel, the polarity has to match the material that you're transporting. So if this is a transport protein for sugars, then this channel has to be polar on the outside. If this is a transport protein for, let's say, lipids, then the lining of this channel would be non-polar. That was it?
Yes. Okay. Thank you.
All right. Of course. Now, whenever I draw the ribosome, we always draw a single ribosome, implying that when the gene is expressed, there's one ribosome and you make one copy of the protein. And of course, that is not the case. Okay. So you're going to read the messenger RNA here to here. Of course, this is messenger RNA. We are going to read the messenger RNA in this direction. So when you read messenger RNA, you read this in what direction? Be careful. This is RNA. You read this in which direction?
5 to 3 prime.
There you go. 5 to 3. Very nice. Very nice. Okay. Because the DNA, of course, the DNA is always read 3 to 5. And since you're reading DNA 3 to 5, the messenger RNA that you produce is 5 to 3. Nicely done. Okay. So then what sits over here at the beginning, of course, is AUG, always. And then over here at the end is some stop codon. There's 3 to choose from, whatever. Okay. And then in between, I'm going to slam more bases in here like this. In between are multiple ribosomes, all reading the messenger RNA simultaneously. By the way, I'm still drawing the ribosomes only 3 bases wide. Understand that's not the case. On Thursday, we will start drawing them their actual size. And so here's my protein. And then here's my protein. And then here's my protein. What do you notice is happening with the length of the protein?
It is increasing.
Sure, it's increasing. As the ribosome reads more and more of the messenger RNA and links more amino acids in a very specific order. Okay. Then, of course, your release factor protein comes in, and that causes dissociation. And what you are left with is your messenger RNA, your large subunit, your small subunit, so this is large subunit, small subunit, and of course your protein. And primary conformation. Cool. Okay. So understand that when we draw this, we commonly put one ribosome here, and then we put one protein here. Okay. But please understand that, of course, the messenger RNA is read by multiple ribosomes. And you make multiple copies of the same protein.
Mr. Memo,
Yes.
for the sorry, does the ribosome like does it not come back together? The large subunit and the small subunit, do they
No.
stay like that?
They stay separate. The small subunit actually has a groove in it, and the messenger RNA actually sits in the groove of the small subunit, and then the large subunit cap comes in and locks the messenger RNA inside of the ribosome, like such. And then when you get dissociation, these two subunits separate.
So, but like afterwards, do the large subunit and the small subunit just float around in the cytoplasm? Are they broken down, or?
They are not broken down. They stay floating around in the cytoplasm. If this is a free ribosome, if this is an attached ribosome, the small subunit stays on the surface of the ER. And when another messenger RNA comes in, then the large subunit floating in the cytoplasm comes back in. So the large subunit floats around in the cytoplasm all the time. Okay. The small subunit, if it's a free ribosome, this floats around. If it's attached, this is attached to the surface of the rough ER. Cool. And then this structure here with multiple ribosomes reading the messenger RNA all at once is called a polysome, or many bodies. So when IB asks you the essay question or the short answer question about transcription and translation, they love to drop this in. Draw a simple diagram of a polysome. A polysome, what's that? It's a messenger RNA with multiple ribosomes. All it is. Sounds fancy, super simple. Now let's make sure, what are the questions? Are there any images on 2E? No images on 2E. Cool. We get 1714, but that's not until 3C. Okay, cool. Comfortable?
Yep.
All right. We can actually hit 3A and 3B very quickly because they are summary questions. And we can leave off 3B or 3C. It depends on how far we feel like getting today. No, copy the questions. I just have. There we go. So 3A. 3A says create a general summary of translation and specifically use the codon chart. So they want you, as I've been drawing the messenger RNA with just tick lines for the bases, they would like you to be able to show that you understand the codon and anticodon. Okay, but all they want is translation. Okay, now when you create this, keep this simple. Okay, so what you want to do is only draw out 9 bases. 1, 2, 3, 4, 5, 6, 7, 8, 9. Oh, the kid's good. Okay. Now, which 3 bases always come first in all messenger RNA of all organisms?
AUG.
Yep. AUG. Bacteria, plants, animals, fungus, everybody starts with messenger RNA. Okay. Which means that of course, you'll put the ribosome here. You'll bring in the tRNA, right? What is the anticodon here? Always.
T.
Careful, this is RNA.
U. A.
You totally agree. UAC, always. UAC. And what is always the first amino acid? Always. methionine, always. Okay, all proteins start with methionine, always. Okay, then pick some codon in the middle for some amino acid, whatever you want, and then pick one of the 3 stop codons here. Pick one, okay? There's 3 to choose from, okay? There's UAG, there's UAA, and there's UGA. Pick one, okay? And then, of course, is there an amino acid here? Let me ask a much better question. Is there a tRNA that comes in here and brings an amino acid? No, what comes in?
Release factor protein?
There we go, release factor protein. You're like, man, we haven't seen this stuff for a long time. Yep. Again, this is about 3/4 of the way through core 2. Okay. And then, of course, this reaches the end, and then you hit dissociation. They are asking, even in the question, it says create a general summary. So what you're talking about here is initiation, elongation, termination. Okay, briefly explain the process. This is that kind of question where you can very, very quickly start writing a full page and just a small paragraph. We're trying to remind you about core 2 and say, hey, remember core 2? All right, remember how you have the start codon, which is always AUG, and then you have the tRNA that comes in, and that is always bringing with it methionine, always. And then the ribosome goes to the bases in the middle. You are going to choose these 3 bases. And then the ribosome goes to the end, and there's no tRNA, brings in a release factor protein that causes this whole complex to dissociate. Okay, brief. Keep this brief. It's just meant to be a review question to lead you into the depth that we're about to talk about. Cool?
Yes.
All right. Let's hit 3B, 3C, and wrap it up. 3B. 3B says the ribosome. The ribosome is composed of a small subunit and a large subunit. Easy. That's why I've been mentioning this all along, so that when we hit 3B, this is nothing new. When you see the ribosome like such, that is a ribosome composed of a large subunit cap and a small subunit base. Just like this, okay? And what you want to do, of course, is label and explain the text images. And you say, which text image? It's 17. This is all in chapter 17. Let's see which one we're talking about. 17, 16. That's what we're looking for. So let's just do 17, 16 today, and then we'll let's stop there. Let's just do image 17, 16. Would that be all right?
Yes.
That'd be cool. Okay. May I take this? Anyone
Yes.
tell me? Yeah. All right. So here is 1716 on page 7 of your notes. There's what a ribosome really looks like. Okay. Now notice that there are 3 grooves in the ribosome. They are called the E, P, and A sites. Okay, E, P, and A. So backwards, it's APE. Okay, so what are we looking at here? Okay, so this up here, this is the large subunit. Like such. Okay, the A site. Why is this called the A site? So let's go for this first. This is called the A site. Why? for aminoacyl. Okay, it's called the aminoacyl tRNA, and I should actually make this bigger for you. You have to see it small. Let's see it big. Boom, there we go. That's much better. Aminoacyl tRNA binding site. But nobody calls it the aminoacyl tRNA binding site. Shut up. Everybody calls it the A site. Okay. And then this down here, this is the small subunit. And here they're kind of showing you that groove that I was talking about, where the messenger RNA dips into a groove in the small subunit. Okay. It's interesting how that black ink shines on there. That's cool. Makes it kind of hard to read there. Is that better? There we go. So this is then the messenger RNA binding site. And then you have the E site, which is really for the exit site. So what happens is the tRNA comes in here, and then the ribosome ships over, and the tRNA is here, and the tRNA is here. So when the tRNA gets here, it's uncharged, and it is ejected, or the tRNA exits. And this will make much more sense when we talk about this more on Thursday. So then this is the P site. Okay, for the peptidyl. You say, what? Because this is actually where the peptide is carried. So this is the peptidyl tRNA binding site. And again, nobody calls it this. We don't call it the peptidyl tRNA binding site. We call it the A site. And we call it the E site, and we call it the P site, of course. Okay. And then what we're hitting on Thursday is this. Okay. So here's your ribosome, right? Here's your messenger RNA. Here's your A site, P site, and your E site. So this is your A, P, and E. And so where we will start on Thursday is how the tRNA comes in here, bonds to the A site as charged, and then as the messenger RNA, as the ribosome slides over, this tRNA ends up over here, and then ultimately it ends up over here uncharged, and then it is ejected. And when the tRNA is ejected, of course, it is uncharged. Notice how the amino acid is missing. Okay, so that's a really good space or place to stop. We will pick this up when we rejoin on Thursday. And if you have any questions, reach out to me, of course, if I can help you at all. And then we will have the TOK meeting tomorrow. And then I will give you an update as to what our plan is for the month of February and developing your essay, because I need that essay by like March 1st, March 2nd. Because then I have to post it to IBO.